Hello students, welcome to Vedantu Neet English. I am Ms. Gopika, your biology master teacher. Please let me know if I am audible, visible and the presentation also is clearly visible in the chats. All of you, am I visible? Yes, I am visible. Am I audible? Yes, all of you.
Yes, ma'am. We are audible and visible to everyone. Welcome to Vedantu Neet English. This is Basavaraj Sir.
This is Gopika Ma'am.
Ma'am is doing again the entire
Yeah.
thing. Students, today's class is going to be very special because, as we promised before, right, ma'am, we'll be doing the entire scientist and important dates from your complete biology. Because you know in your examination they will give you direct question that who is a scientist who worked on this experiment, or they can ask you important date directly. Do you require a lot of thinking here? No. If you know the date, if you know the scientist, easy 4 marks for you. So this is a session which will help you to gain those 4 marks. Right, ma'am?
Yes, yes. Okay, audible, visible. Okay, awesome. Fine.
Yes.
Okay, so you know what you should do when we start the class? Only Subhashini has done it. Others are absent in the class. Okay.
They're not present. They're not present here.
Yes. You cannot be all inactive because we are getting closer to Neet and you need to be hyperactive now. Okay, because all the classes, so much to, take it
Yes.
in. Right? So we are making it more easier for you by making the PPT and just sharing it to you. Imagine you have to sit and write this. It will take you a decade. You'll
Exactly.
keep on,
It requires
Break.
a lot of effort to students quickly like this. I want to smash that, like the smash button.
smash the like
Smash the like button quickly right now, and we'll start with the all the scientists. Yes.
Okay.
And don't
Yes.
worry, the entire PDF will be available for all the students who are commenting, sir, PDF, sir, PDF, sir, PDF. Don't worry, the PDF will be available. Yes, sir, Sharad Jasni, yes, the PDF will be available to you on the Telegram channel. Right?
Yes. Okay. Awesome. Shall we start?
Can we start, students?
Yes, we can.
Well, they're asking, how are you, ma'am?
I'm good. I'm awesome.
So much of care.
I'm awesome. Yes.
Ma'am, the first scientist, ma'am.
Okay.
Now.
Oh
Yes.
my god, this is a very important question.
Yes, students, before you start the chapter, before you start any unit, there will be some scientists will be given before every unit in your NCRT. That is the first, before start of the first unit, they have one scientist called as your Ernest Mayr, who is known as the Darwin of the 20th century. Now, what are his contributions? Look at the first one. That is his research. His research spanned from ornithology, taxonomy, geography, evolutionary and systematics. That is the entire systematics. The binomial nomenclature, like even after the Carl Linnaeus, he was the person who worked on your taxonomy, zoology, as well as your embryology also. Students, Ernest Mayr, Darwin of 20th century, you need to remember this line right here. This line right here, please remember that. Okay. The next one, ma'am, that is going to be your Carl Linnaeus. Everyone knows Carl Linnaeus, how important his contributions are towards the development of taxonomy, right? As well as the binomial nomenclature.
Even the book.
Even the book, exactly. So the generic, so he gave the, each
Generic.
name has two components. One is your generic name and species epithet. Ma'am, this right here is a Neet PYQ.
Yes.
And it was probably like around 2 years back. Right? 2 years back, this was a PYQ. And this is
Yes.
called as binomial nomenclature. Clear?
Very remarkable, he changed the whole thing. He made it very, it made things to be labeled easier. It's not
Exactly.
like Basva sir in Karnataka is finding a plant and he's calling it, okay.
Random name.
Yeah, he's calling it Champa, and then I am sitting in Andhra and calling it Pandu, and I'm like, okay, we both discovered a thing and we both are trying to publish it. So now
So-
what happened? Patent issue will come, right?
Exactly.
All of you with? Yes, book is Systema Natura.
Then, ma'am, we have George
Bentham and Hooker.
Bentham and Hooker.
Okay.
So
John
Ben-
and all of it, Joseph
difficult.
and John and all.
So Bentham and Hooker classification. So students, there are multiple type of classification, but primarily which we use in your taxonomy, especially in case of botany, is Bentham and Hooker classification. That is natural system classification. Now we have artificial. Artificial mainly uses the external morphology. But when the true love is, ma'am, true love is you look at inside a person, right?
However,
However
However, a person-
beautiful, however beautiful a person is,
does not matter.
doesn't matter.
Heart matters.
Heart matters, right?
Okay.
The circulation matters, right?
Yeah,
Internal. Soul matters. That's what Benjamin Hooker did. Instead of looking at the external morphology of the plants, they looked at the internal structure of plants. That is anatomy. Internal, which is like your ultrastructure, anatomy, embryology, as well as your phytochemistry. The phytochemistry basically involves the metabolites, primary metabolites, secondary metabolites, and all of those. So remember, students, okay? That is your Benjamin Hooker classification. Now, next, before we go, students, before we go to the next scientist, let me quickly tell you about the Vidya Utsav, which is happening on the 1st May, right, ma'am?
Yes. So some very, very, very huge news is coming, and it will be such a happy news for all the 2025 aspirants and also for the 2024 aspirants, because you people are very close. May 1st, and then only you have 4 days, not even 4 days, 3 days.
left.
Okay, 3 days. So very exciting things, and something that you have never seen from your teachers end. Something you know
Yeah.
outside the box. You cannot like
Even imagine it.
yeah, you can imagine it. Fun activities means that. So join. The link is in the description. Okay.
Yes, ma'am. Ma'am, next scientist, ma'am.
Yes.
Again,
Carl-
students, Carl Linnaeus, again, that is your artificial
Artificial
system of classification. Carl Linnaeus is mentioned in multiple chapters. That's how important he is, you know.
Yeah.
Next one is Aristotle, ma'am.
Aristotle did two kingdom classification system, which ultimately failed because he only put plants and animals. And again, plants, and before putting this plants and animals also, there were a lot other people who put no, depending on the red blood cells,
Exactly,
depending
ma'am.
on trees, herbs and shrubs. Okay, so Aristotle did that, and after that, students, actually here we can write Carl Linnaeus again here also.
Yes, Carl Linnaeus also.
Carl Linnaeus. This Aristotle did the classification depending on the presence and absence of red blood cells and trees, herbs, and trees, which completely failed.
Herbs, shrubs, and trees.
Trees, yeah.
That is based on the stem length as well as the.
Yeah, just looking at the plant, the habitat.
Just look at the road and you'll be like, okay, fine, this looks very short, call it herb. Okay, this was a little bit taller, call it shrub. One more taller, call it tree.
And
That's
then
all.
Carl Linnaeus came up with two kingdom classification, which he classified plants and animals. This made a lot of sense, but again he forgot to put eukaryotes, prokaryotes
Prokaryotes,
separately,
nothing
photosynthetic, non-photosynthetic. Everything completely failed, but it is important for you to know the feedback, right?
Yeah.
What is the drawbacks of that?
Like in chemistry, they learn, right? The
Yeah.
J.J. Thomson model, why it failed, then we have new model. Similarly.
Now, the most important one is R.H. Whittaker's classification. It was in 1969, and this is the most widely accepted classification, your five kingdom classification. All of you in the chats, tell me what are the five kingdom classifications. Go start.
Go in order, go in order.
I'm giving you clue. Monera. Start with Monera and then go for it. Okay.
Go with Monera, then we start one by one.
Yes, quickly. I want the answers quickly, and I also want likes. And if you're new to the channel, do not forget to subscribe, because until I tell you, you people won't do. You work on artificial intelligence. Okay.
They are artificial classification, ma'am. Unless we tell them, they will not function only.
You people don't love us from heart. That is what is proven. Yes,
Yeah.
very good. Come on.
Yeah,
Nobody is telling something.
Just say Monera. Okay, I don't know rest. I just say Monera and I'm like done.
Come on, only Monera. Continue.
Yeah.
Very good. All the five you should be in a
All
position
five.
to tell now.
Exactly. See,
Very
Subhashini.
good. Subhashini. Yes, awesome. Monera, Protista, Fungi, Plantae, Animalia, most widely accepted classification. Now, your Ivanovsky, okay? Dmitry, I'm not telling. Okay,
It sounds like a DJ name, right?
Yeah, DJ.
DJ.
So, yeah, Ivanovsky in 1892, very earlier, he recognized microbes causal. That means what does it cause, right? So, microbes causal organism of tobacco mosaic disease. That means he was actually after, he was trying to find tobacco mosaic virus disease. How does it come? Okay, so the causative agent of it. So that's how he came to a recognition that certain microbes are working on it. Okay, awesome, fine. Then, this next name, Vejrick, okay, M.W. Vejrick, again in 1898. Okay, what did he do, students? He basically extracted the infected plant of tobacco and could cause an infection in that healthy plant. Okay, and he named it a new pathogen called as virus. So these two people were given the recognition for finding virus. Simple as that. Okay, and he called that fluid as contagium vivium fluidum. That means something that could be contagious or infectious. Okay? Now, okay, again, Stanley.
Yeah.
Stanley also very famous, important person.
M.W. Stanley in 1935, he showed that the viruses could be crystalline and crystals consist largely of proteins. So it gave us an idea what the structure protein is. Similarly, in your DNA structure also, we did a crystallization of DNA. Here we have a crystallization of your viruses, which mainly contain your proteins. That important point we should remember. And they are that is inert outside the host cells. Viruses are obligate parasite.
Only if they enter the host cells, they are
Be
active.
active.
Otherwise,
Outside,
they are inactive, right?
active.
So that also finding came from Stanley. So it's very important. Any one point among this, if they ask and they ask
Directly.
you Stanley, like incorrect or correct, you should be in a position to tell. Don't be like, oh, Stanley told it's crystallized. So
Yeah.
only for that word I will find.
No.
Okay, got it?
They'll,
Yes.
they'll not even think twice, ma'am. They'll just say viruses and then they'll go to the venue.
Yes.
Yeah.
Now, Denia, 1971, Viroids. Viroids are smaller than virus but very, very infectious. This was found. Now, students, date is very important, okay? 1971. This date as such is asked. Other than that, the scientist name also is
Has
asked.
been asked.
Okay?
Ma'am,
Yes.
then we have again it's repeated, so let's just skip this one.
Yeah.
Then Catherine Sau.
Yeah.
Catherine Sau.
Students' names are from directly, I don't know, it's not even there in Oxford.
The thing is, ma'am, they're not, most of the words are not even English.
English.
They're
Yeah.
Ukraine names or they're name from the country, so Italian or the
So
pronunciation
please
can vary.
forgive us because we are
Ah.
Indians. Our pronunciation will be very Indian.
Yeah,
And
Indian.
don't come in the comments and be like, oh ma'am, you chose this instead of this. We are
It's
all
fine,
Indians.
Indians. We can make mistakes.
Only thing you have to remember is that fellow's name and the anyway.
Yeah.
Thank God you do not have spellings because I can imagine what you would have done with these.
Oh
names.
my God! Imagine
Okay.
writing the spellings they have gone.
They would have written something and all. Okay.
Okay.
Dates are important. Knowing the names are important. Okay.
Now, she reported that in a early publication that curly top virus spread through the plant via food conducting or phloem tissue. Now, if you ask me who is one of the most pioneer in your entire plant anatomy, this is the name. Remember the plant anatomy. This is the name which you need to remember. And in 1984 took a dynamic development approach designed to enhance one's understanding of plant structure and enormous impact of worldwide. That is, how does the internal structure of the plant works? Catherine Nessu was the person who discovered it. Remember that. Clear?
Exactly. Nobody.
Ma'am, SO is not in NCRT, right? No, it is given in your NCRT. If you, before your entire unit starts, before your structural organization unit starts, you have a dedicated page for Catherine SU. Don't be like, no, it is not there at all. It is there in your NCRT and they might ask you from there. It might not be there within your chapter, but it is there in your NCRT before the unit starts.
Okay, so I also want to tell this in this student Stanley, Denia, the previous slide, all these people know some. I noticed that in the old NCRT it is maybe a scattered or some names are missing, but in your new NCRT there's a dedicated paragraph to explain all of this. That is your five kingdom classification. You forget from here, okay, from Ivanovsky and your Bedrick and your Diner, then Stanley, all these people, there is a dedicated paragraph. I don't know how many of you have read your NCRT, new NCRT. You can go to biological classification, check. There's a paragraph. So don't get confused. Go to Google, download biological classification new PPT and you can learn it or learn it from this PPT, the PDF that we are sending.
And it doesn't require much of, imagine, you don't have to read the entire paragraph. Just remember the names and their main contribution.
Yes.
See, like I think as I told you, it is, you have worked on your virus. Right, virus also, and anatomy, main function. That's all you need to remember. Extra, don't need to remember. Just few points which we have marked here for you. Remember these points and you're set. Right now?
Okay, done.
Next one. Then we have G. N. Ramachandran. All of you would have heard about the protein structure, right? The protein structure was first elucidated by a Ramachandran plot. Here you need to remember what two lines here. That is, the first point you need to remember here is the triple helical structure of collagen. This is the first point which you will remember about Ramachandran. Triple structure of collagen. First point. The second point which you need to remember here is the Ramachandran plot. That is his analysis of the allowed conformations of protein through the use of Ramachandran plot. Now, what is Ramachandran plot? Do you need to study that? No. You do not have to study Ramachandran plot. But do you need to remember Ramachandran plot was given by whom? And also one more point here. Yes. So these are 2 points you need to remember. Again, Ramachandran is not given in your chapter per se. Here, Ramachandran has been given before your cell chapter starts. Okay. Now,
Anthony Van Leeuwen.
and beyond Leeuwenhoek. So first describe the live cell. So first living cell was observed by Leeuwenhoek and with the help of microscope.
Yeah, the Koch set. Right?
Then we have the Robert Brown, the discoverer of nucleus. Year very important for this. For this, year is very important. Now, the next two scientists for your cell chapter are going to be very, very important. The two things are your Scheiden and Swan. Scheiden and Swan, the two scientists who gave the cell theory in the initial stages. What did Scheiden do? Scheiden was this line, ma'am. Here, students often forget this. He was a German botanist, while your Swan was a British zoologist.
Can you
Now,
see the combination? How nice it works. It
Yeah.
has done miracles. Okay. Yes. Fine.
right, these two lines here, German botanist as well as British zoologist, man, they can ask you directly. They will not give you Scheiden and Swan name. They might give you German botanist who discovered this, right? Cell theory, exactly. So examined a large number of plants. So remember, obviously botanists will work on your plants and observe that all plants are composed of different kinds of cells. That is, your entire plant is made up of many different types of cells, and that was given by your Scheiden. But Swan is a zoologist. He will work on your animals, obviously. So British zoologists studied different types of animal cells and reported that cells had a thin outer layer. A cell has a outer layer called as plasma membrane. So plasma membrane was first identified and discovered or elucidated by your British zoologists. Back then, they did not know what is it because of there was no advancement in microscopy. But later stages, they were able to tell. Yes, it is plasma membrane. Now together, ma'am, your Snyder and Swan, they give a cell theory, telling that every cell, both of us, ma'am, we are organisms, we are made up of cells and parts of cells or products of cells. But one thing was missing here. That is, how does cell come to origin? Like, where did we get the cells from? That answer was given by one more scientist called as Virchow. It's given by Rudolf Virchow was the final scientist who gave the last piece, missing piece to your cell theory. He finally told us that the first explained that cells divide from new cells are formed from pre-existing cells. So whatever new cells you get are formed from your pre-existing cells, and he called it omnius cellula e cellula. Every single cell is from your pre-existing cell. See, that are the 2 points of your cell theory. First point, cell or organs are made up of your cells and products of cells. The second point is cells originate from pre-existing cells. No, this one. Very easy, man. Singer and Nicholson.
Singer and Nicholson.
These two dates are very important again.
So there it is 1838, 1839, 1855. Cell theories order. Okay, how the scientists found one thing. Okay, now Singer and Nicholson in 1972. Okay, fluid mosaic model. Fluid mosaic model led to a complete discovery of your lipid layers. How is the bilipid layer there? How is the
protein movement and everything?
Yeah, hydrophobic, hydrophilic tail, intrinsic protein, extrinsic protein, and much more. It's a beautiful topic by itself. But then, as of now, you only need to know fluid mosaic model. Are you all with us?
Yes.
Are you all with us? Can I have some red hearts and green hearts in the chat box, because I cannot feel the energy. I feel like all of you are chumma sitting and talking with each other. Did you eat breakfast? Did you eat lunch? You're not talking to us.
Yeah.
Okay.
Talking to each other.
Yes, you should be able to answer. The moment we tell Singer and Nicholson in the chats, fluid mosaic model should be the answer, students.
Yeah.
If not now, then when? When are you going to be serious? Okay. So quickly, come on.
Let's go next one, ma'am. Camillo Golgi again.
Yeah, Golgi.
Discovered the its nucleus. No.
I think that got.
This is supposed to be discovered the Golgi body. Okay, Golgi apparatus.
Yes, students,
Okay.
from the name only you can understand.
Uh-huh.
Okay. Next one is again George Palade, ribosome.
Ribosome का organelle structure in 1953.
53. Okay.
Then we have Melvin Calvin. Now this sounds so nice. Melvin Calvin. So Calvin cycle. That is your C3 cycle. Right. Calvin proposed that the plants change light energy to chemical energy by transfer of electron in an organized array of pigments, molecules, and other substances. And in a dark reaction, glucose is being formed.
So when sir is telling this, array of pigments, in your head already you should be able to see the pigments, and you should be able to see the flow of electrons.
Yeah, like Z scheme should be seen and formation of C3 cycle should be happening.
Yes.
Next one.
Okay.
Joseph Priestley.
Again, very beautiful scientist. Again, did a very revolutionary thing
Beautiful,
in
no?
17. Too early this is, okay?
Yeah,
In
very early.
1770. Okay? 1770 again.
So I will not be explaining the entire Bell-Jar experiment. All of you know Bell-Jar experiment. But one more contribution. Apart from the Bell-Jar experiment, he was the person who actually discovered your oxygen.
Yeah.
If you did not discover, you would not be able to breathe now.
It's alright also. Okay? Yes.
Joseph Priestley discovered oxygen. Again, date is very important.
Okay. So there it was 1770 and here it is 1774. Okay? Yes? Very good. Awesome. Then again, your Indian house, about your experiment, the small bubbles because of the aquatic plant kept. Hydra was used?
No, for this there was the
What is the plant?
Cladophora.
Yes. Okay. I
Indian
think they
house.
won't ask that, no? Do they have the name?
No, they not ask that.
Yes.
So that is when plant was kept here. When the plant was kept and wherever the bubbles were formed means gases evolved.
Did I say Hydra? I'm such a zoology teacher. Hydrilla was in my head, but I told Hydra.
And one more thing you need to remember here is the Joseph Priestley. It's a whatever experiment Joseph Priestley did, ma'am. He same thing he copied.
Yeah. He did basically the same thing, but he just used an aquatic plant there.
Same thing he copied it and aquatic.
This guy used mint plant. No?
Mint plant. Yeah. But he changed it, same experiment. It's like same copy the homework but change it a little.
Yeah. It's like in maths class they'll give 72 into 12. We will do 74 into 12. That's same thing. Okay. Again. Sack. You should remember it as a sack experiment.
Junior small sack provided evidence for production of glucose when plants grow. So when the plants are growing, glucose is formed that was given by your sack.
So how I remember this, glucose is stored. Where do we store it? In the rice sack. Like rice and all, we store it in a rice sack to one sack. Okay, again students, 1854. So you can see from 17, only 17 I think we studied this year, right? Then 18, 2 dates we have studied. Then Sir Engelmann, because it's Engel, you can remember it as Engelmann. Okay, he's the one who formed some angles and proved it. Okay, so again, a first action spectrum of photosynthesis was just described.
That is, remember your entire prism experiment. The
Engelmann.
Entire prism experiment, scattering of light, and at which particular wavelength, which particular pigment is being absorbed. All that was given by your T.W. Engelmann. Now here one more thing you need to remember is someone is saying, sir, ma'am, you can use identify word. We are not making this a discovered word. NCERT is giving it.
What?
Instead of writing discovered, you should write identified.
Oh,
it is technically correct. But NCERT is telling you discovered, so we are using discovered directly. Then we have Cornelius Van Niel, that is your H plus donor is hydrogen. That is, if you want to reduce your CO2 to glucose, right? CO2 to glucose, we need hydrogen. Yes, do you need hydrogen? And the hydrogen is from your water. Hydrogen is from water. And that was given by your microbiologist Cornelius Van in 1770. Again, way back ahead. And remember students, Cornelius Van Niel did his major experiments on your purple and green sulfur bacteria. That can also be asked as a question. Okay. Now, next one is going to be a Kranz anatomy. Now here I didn't want to mention this, but it's very important if you want to understand Kranz anatomy and how C4 plants work. See, in case of your C4 plants, that is, Kranz means breadth and the reflection of the arrangement of cells. The bundle sheath cells, see, in the middle we have the bundle sheath cells. Bundle sheaths are completely covered by your mesophyll cells on the outside. So in the middle we have your vascular bundles. In the middle we have the vascular bundles. Then we have bundle sheath cells, and then we have mesophyll cells. And bundle sheath cells are so tightly packed that nothing can enter inside, and they have a higher number of chloroplasts. Then we have hatch inside pathway, that is nothing but your C4 cycle. And all of us know what C4 cycle is. We have two different types of cells there. One is a mesophyll cell, other one is a bundle sheath cell, and how the glucose is being formed.
So I'll ask one question to your students. Students, C4 cycle is better for the plants or C3 cycle is better for the plants?
Simple question, students. You should be able to answer this question.
Which is better? Which was better in the case of Rubisco, in case of many things, affinities towards carbon dioxide, oxygen. I think I do too much favoritism to students. I should just stop I should not give hints. Come on, tell me C4 or C3.
Exactly.
Both has its own drawbacks, but then still, okay? Still, which is one?
Students, you know that C4 ma'am always tells C3 plants are stupid plants. C3 plants are so stupid just because someone else is coming, they're leaving their partner and they're going. Just because Rubisco found oxygen, it's leaving CO2 and it's going. So that's why C3 plants are stupid little. C4 plants are very smart.
Yes.
But if I'll tell you, I'll ask you one more question now. C3 plants or C4 plants, which plants can grow in more CO2 concentration? Who has the higher number of saturation? Is it C3 plants or C4 plants? Which plants has more CO2 saturation? That is a good question now.
Come on, answer. What if this comes this time for
Yeah.
Neet 2024 in a different way? The students definitely half of the questions are basically change in grammar. They just active to passive, passive to active. Okay? Okay, they're still trying to say. Yes, maize, sugarcane, C4
Exactly,
plants.
C3 plants, ma'am.
Yes,
For
they're
your
saturation.
saturation C3 plants, because C3 plants, that's why C3 plants are grown in your greenhouse.
Yes, awesome.
Example,
Okay.
tell me all the examples. Tell me. Example is also very important for that.
Yes.
Then we have, ma'am, MDen manner of pathways. That is basically your EMP. That is scheme of oxygen. Now, students, tell me what is this? I'm gonna ask. Students, what is this EMP pathway? I don't know. I don't know what is this EMP pathway. Can anyone tell me what is this EMP pathway now? Tell me quickly. What is EMP pathway? Then we have Charles Darwin and his son called as Francis Darwin. Both of them were involved in discovery of oxygen in your canary grass. That is photoperiodism. Then we have isolation. That is by F.W. Hunt. F.W. Hunt did the isolation of your oxygen in Avena sativa. That is Avena sativa curvature test. So remember, discovery was of oxygen by Charles and Francis Darwin. Isolation of oxygen by F.W. Hunt. Students can go wrong here big time.
Yes.
Confusion. Then we have your Kurosawa. It's a Japanese guy, Kurosawa. He identified a plant called as your type of your rice plant, which is growing so tall. Ma'am, this plant is growing so tall, but there are no seeds in it.
Okay.
He called it foolish disease. And this foolish disease was caused by a fungus called as your Gibberella fujikorai. That all sounds Japanese, no?
Yeah, Fujikorai.
Fujikorai. Gibberella fujikorai. That is discovery of gibberellins, 1926. Again, very important. Then we have Skoog and his workers, discovery of cytokinins. Then we have Miller et al. That is his worker, friends. 1955, identifying crystalline of cytokinins and promotion of active substance that are termed kinetin. And this was done in your herring sperm fish. The fish, there are herring fish. Sperm was isolated. So remember, discovery of cytokinins by Skoog, isolation by Miller. Again, discovery, isolation. Then we have H.S. Cousins. Confirm the release of your volatile substance from the ripe orange. That is basically your which gas? Ethylene gas, which is the only gaseous hormone. So this is your ethylene. Your ethylene. Ethylene was discovered by H.S. Cousins. Then we have Panchanan Maheshwari. Now you might have been like, sir, I do not know what is Panchanan Maheshwari. I've never heard of this scientist. Again, students, this is one of the scientists which is given in your previously before your unit starts. And remember, Panchanan Maheshwari, that he worked on embryological aspects and popularized the use embryological characteristics in taxonomy. Very important. Embryology. So Panchanan Maheshwari, please remember that. Gregor Mendel.
Okay, Gregor Mendel. Okay, you people all descended Mendel, but yeah, Gregor Mendel again, awesome person. He did hybridization experiment and 7 pairs of contrasting traits. That's your P plan. All of you know that's how your monohybrid, dihybrid, cross, and everything about principles of inheritance came from.
Law of dominance, law of segregation,
yeah.
law of segregation, law of independent assortment, as I was saying, right?
Yeah.
So all of this were given by your Gregor Mendel.
Yes.
Clear? Remember the laws which is going to ask in examination. Remember the incomplete dominance also. Incomplete and co-dominance both are very important. Okay.
Yes.
Then we have Hugo de Veris, that's from your ma'am, if I remember, he was there in evolution also, right?
Yeah. Hugo
de Veris.
Mutation.
Right? Karl Kohns and Schumacher that independently rediscovered the Mendel's result on the inheritance of characters. So students, remember, Mendel's work was completely ignored for such a long time because they were like, who is this person using mathematics in your biology? They're like, you tell me, you yourself don't like mathematics, right? When it comes to in biology, how can other people also like? They were like, no mathematics here. They were like, no mathematics. So his work was completely ignored. But later on, 3 scientists rediscovered his work in 19s. Then we have Certain and Boveri. All of you know Certain and Boveri, chromosomal theory of inheritance. They basically tell you that chromosome and gene has a similar parallel behavior. That's it. Not going to go deep into it right now. Then we have Thomas and Morgan, that is experimental verification of chromosomal theory of inheritance. So they can ask you directly, chromosomal theory inheritance was given by whom? Experimental proof was given by whom? Theory was given by certain Bovary. Experimental proof was given by your T.H. Morgan. Now here, this is a P by Q at least 3 times. 3 times they have asked you. That is, T.H. Morgan had a student. His student is Alfred Stuartman. Now, what did he do? His main work was on your gene mapping. I'll not read this right now. So you should remember his main work was on gene mapping. He told us that on the chromosome, which gene is present where. That was given by your Alfred Stuartman. Remember, student of your Thomas Morgan. Then we have Henking. Henking in 1891 gave the X body. So his main work was on sex determination. Sex determination. In sex determination, he studied that 50% of sperm do not have a nuclear genetic material. 50% of other sperms had something. Whatever was missing, he called it X. And we know it as X chromosome right now. Then we have Punnett. All of you know the famous Punnett square. Easy, nothing to know that Punnett is Punnett. Then we have Langdon Down, who gave us the Down syndrome. Friends, quickly tell me in the chat. Tell me in the chat quickly. What are the main symptoms of Down syndrome? And also in Down syndrome, do we have extra chromosome or do we have one chromosome less? Tell me that also.
Yes, come on, all of you. Yes, numericals will definitely come from principles of inheritance. Sir has already done numericals.
I have already done so many numericals.
Yes, sir has already done numericals in the many sessions we have done. So just go watch the session because we have done as many questions as possible. Okay? Yes.
Then we have again Herbert Boyer. They have not given you this in the chapter. They have given you before the biotech unit starts. They have given you it. That is Herbert Boyer. Boyer observed that there are certain enzymes that have capacity to cut the DNA strands in a particular fashion, which left known as sticky ends. That was your restriction endonucleases. Then we have Stanley Cohen. Stanley Cohen has been studying the small ringlets of DNA called as plasmids. So remember, Boyer main work was on restriction endonucleases, while your Cohen main work was on plasmids. But when they came together, they had a bigger work. The bigger work that was they were first to demonstrate rDNA technology. Remember the experiment, Salmonella. We took out an antibiotic resistance gene from your Salmonella and we added it to your E. coli. The first time we ever made rDNA technology, it was because of your Stanley Cohen as well as Herbert Boyer in your 1972. Again, dates are very important. 1972, first time rDNA technology. Okay. Then in year 1963, again very important. What happened in 1963? Two enzymes responsible for restriction in the growth of bacteriophages were discovered. Two enzymes. One enzyme was called as what? The first enzyme was called as your restriction endonucleases, which is going and cutting your bacteriophages. And the second thing was your methylation. Second one was your methylation. Okay. Now, next one is your Bolivar and Rodriguez. All of you know that is the scientists who first constructed your PBR 322. P for plasmid, B for Bolivar, R for Rodriguez. 322 is basically the order of discovery. Clear order of discovery. So PBR 322 cloning vector. Then we have Ramdev Mishra, again very important figure. Now if you look at your entire ecology, go search your entire ecology, you will not find Ramdev Mishra. Why? Because his name is given before even the entire chapter starts. And do you know who is this person? The great scientist. He is called as the father of Indian ecology. Right? His research laid the foundation of understanding of your tropical communities and their succession, environmental responses of plant population and productivity, nutrient cycling in tropical forest as well as grassland ecosystem. So if you look at your entire ecology, the Indian ecology was given by your Ramdev Mishra. So remember the name, don't forget. Okay. Then we have Gauss principle. Gauss principle is very simple. Computative exclusive principle basically tells you that when there are two individuals staying in the same area fighting for the same resource, what will happen? The inferior one will completely perish. The dominant will survive. Okay, dominant will survive. Next one. Now,
Students, a lot of scientist names comes from your, again, molecular basis of inheritance. So first one is your Friedrich Mischer. After this, you can remember Altman also, but Altman is not mentioned in your NCRT. Okay. So the first person is Friedrich Mischer in 1869. DNA. He found that DNA had some acidic substance. Okay, DNA had some acidic nature, and because of it, he called it nucleic acid. Now, all of you tell me in the chats which are the two nucleic acids that you have studied in molecular basis of inheritance. Then, students, your James Watson, Crick, Rosalind and Franklin, all these people with the help of X-ray diffraction was able to find how DNA looks, and that came to the discovery of double helical structure. But the catch here was only 3 people was awarded Nobel Prize, or only 3 people were awarded. Franklin was completely ignored, right? Yes. Awesome, awesome. Which is the nucleic acid? It's DNA and RNA. Okay, students, Altman also is here. You can also write about Altman. Okay, the discovery of nuclein. Okay, the discovery of nuclein. Again, here, remember Watson, Crick, Wilkins, and Franklin in the year 1953 with the help of X-ray diffraction ended up finding double helical structure. Now, Chargaff in 1950. Students, the date is not mentioned in NCRT, but I just did research, and it is showed that in 1950 Chargaff came up with the Chargaff rule that proved that always purine has to be equal to pyrimidine, or the ratio of purine should always be equal to pyrimidine for the double helical structure to be maintained, or for the DNA structure to be maintained, because again structure was still being found. So he just told that this keeps the DNA in position. Okay, clear? Yes. So Chargaff rule, that is adenine is always equal to thymine, or not like that. Your purine is always equal to pyrimidine. How much adenine is there, that many thymine should be there. How much cytosine is there, that many guanine should be there. Okay. You can remember it as at Gopika Ma'am's class. All this PDF you will be getting on Vedantu Neet English Telegram, as well as on Biology by Gopika and Basavaraj Sir's Telegram. So you can quickly join our Telegrams and get the PDF. Okay. Now, next is Griffith. All of you know Griffith found out your transforming principle in the year 1928. He did not know what was the thing that got transformed from the S train to the R train, but he just called it as transforming principle. Okay, transforming principle. Okay, now after that, many other scientists or many other zoologists and botanists came after the transforming principle, and one or three among those people are Avery, MacLeod and McCarthy. They came up, or they were trying to find the biochemical nature of this transforming principle by using protease, RNase, and all of you in the chats, protease, RNase and one more enzyme they used to find the biochemical nature of transforming principle, but it did not get accepted, and it went almost for 10 years, 1933 to 1944. Okay, so if any of these dates come, you can mark the dates, the initial and the ending date, right? 1933 to 1944. Which is the last enzyme that they used? Protease, RNase, and quickly. Yes, Avery, MacLeod, and McCarthy. Hello, Shruti. Yes, okay, awesome. Fine. Hershey and Chase in the year 1952 proved that DNA is the genetic material, or the only experiment that could prove that DNA is the genetic material with the help of bacteriophages, E. coli by infection, and using radioactive substances, right? So this experiment is widely accepted, and it was in the year 1952. Okay? Yes, very good. DNase was the last one. Fine, awesome. Now, next one, students, Messelson and Stahl. Messelson and Stahl in the year 1958 were able to find that DNA replicates with in a semi-conservative manner. That means that is conservative, dispersive and semi-conservative. Out of these three techniques, our DNA replicates in a semi-conservative manner was proved by Messelson and Stahl experiment. If all of you can remember your N14 and N15. Okay. Now, next is George Gamow. Most of you will ignore this scientist or ignore this physicist because his name comes in a big paragraph of your genetic code. So when you're reading it, you'll be like, you'll ignore this person, but he's very important because he's a physicist. He was not a botanist or a zoologist. He came up with a theory telling that for 20 amino acids, there are only 4 nucleotides. Now, how do we use this? We have 4 this side, we have 20 this side. So they came up with different maths, 4 into 1, 4, okay, or 4 to the power 2. They were like, okay, 16. 4 to the power 3, they were able to find 64. And with the help of 64 codons, they were able to code. Or they were like, okay, 64 codons are found. We have only 20 amino acids to code. So Gamo gave this revolutionary thing, and that's the reason how your translation happens smoothly. Are you all with me on this? And that's how they also prove that codon is triplet. Okay, so here I've written in a combination. How can we, with the help of this 4 bases, find a combination? Okay, that is 4 to the power 3. Then your Govind Khurana, again a very Indian person, right? Most of you will like the name Khurana. We keep telling, no? So yes, Hargobind Khurana, he was very helpful in synthesizing RNA molecule with defined combination of bases like homopolymers or copolymers. Okay, again a kutti paragraph is mentioned after your genetic code where multiple scientist names have come, which most of you will again ignore. So he came up with homopolymers and copolymers. Okay, next students, your Marshall was the person who came up with the cell-free synthesis system for protein synthesis. Finally helped to code. Okay, code to be deciphered. Okay, deciphered. So again, you don't have to know in detail, but you just have to know he came up with a cell-free system. Okay, cell-free system. Yes or no? No, you don't have to study the triplet codon. You don't have to study the whole box. You can remember some common amino acids like lysine, valine, and this AUG already you know. So phenylalanine. Some few of them you can remember, which has been completely repeated in your textbook in else places. You can know few of them. Okay, got it, Madhumita? Okay, now students, cell-free system. Again, your Marshall. Then Severo Ocho. He was the enzyme name itself is Severo Ocho enzyme, but the scientist name is Severo Ocho. Okay, don't get it confused. In your textbook, it's given Severo Ocho enzyme. That is your polynucleotide phosphophoride. Why am I not able to tell? Phosphorylase. Oh my god, got it. Okay, polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in template independent manner. Okay, so basically he found an enzyme and this was the enzyme and the enzyme was called after his name. Okay, are you all with me on this? Again, this is also mentioned in a small kuti line in your textbook. Just know this. Okay, just know at least one term. So anywhere you will be able to find, you will be able to answer it. Got it? Chalo. Next one, Edward Wilson, biodiversity.
So Edward Wilson was a person in the 1891 biodiversity, the term. So if you look at your NCERT, your ecology especially, there are many scientists who came across and he gave you so many terms. The first one is going to be your Edward Wilson. The next one is your Robert May in your 1866 global species diversity at 7 million. So Robert May told us that the entire biology, the entire globe has around 7 million species. Okay. Then in the one more date is very important. That is in 2004, according to the International Union Code for Conservation, that is IUCN, the total number of plants and animal species described so far are slightly more than 1.5 million. So Robert May is saying 7 million, but according to your IUCN, we have around 1.5 million species right now. Then we have Alexander von Humboldt who gave the species area relationship. Now again, they can ask you what is species relationship. That is, the more you search, more species you will find, but only up to a certain limit. That is your species relationship. Now again, here what is important is this equation right here. Log S is equal to log C plus Z log A. Now they can ask you any of the following. They can ask you what is Z, what is C, what is S. Now quickly tell me in the chat, everyone. Quickly tell me in the chat what is the meaning of Z. What is the meaning of Z? Okay, what is the meaning of Z? Then we have David Tillman. David Tillman gave us the Tillman found that the plots within more species. So basically, he told us that in an area, more species are there. So if more species are there, more resistance will be there. Okay. Let us show that in this experiment, increased diversity contributed to higher productivity. This line, that's all you need to know. This one line here. That's all you need to know. Okay. Then we have the Paul Enrich. He gave the Rivert Popper hypothesis. Now, what is the Rivert hypothesis? He basically compared your entire ecology to an aeroplane. He told us that an entire aeroplane is an ecosystem. Now, if you in the aeroplane, if you remove one particular nut or rivet, one rivet if you remove, will the aeroplane sustain? Tell me. Yes, it will sustain for some time. But imagine if you take out around 100 rivets or 200 rivets, the plane will not fly anymore. There is a chance that plane might, crash somewhere. Similarly, imagine you take a rivet out of the wing or of the main engine. What will happen? The entire airplane will collapse very fast. So that gave us an idea about how a species, like disappearing of one species, okay. But if that happens for a very long time, imagine one species keep on removing, one more species keep on removing year after year, and at one point we might face a situation that we do not have any species left. Your entire ecosystem will collapse. And in the entire ecosystem, there are few species which are very important. We call them keystone species. Okay, keystone species. Then in the year 1962, so 1992, we have the Earth Summit. Again, very important. The Earth Summit, very important. Earth Summit held in your Rio de Janeiro. Many questions have come here. Many questions have come. They will either ask you the year. They will either ask you the year. I'm telling you, this is an EPYQ. Either they'll ask you year or the aim. Now, what is the aim here? All nations to take appropriate measures for conservation of biodiversity and sustainable utilization of benefits. This is the aim which they have been asking, or they'll ask you the year, or they'll ask you the Rio de Janeiro, the location. Similarly, we have one more. In 2002, we have the World Summit on Sustainable Development, where in the Johannesburg, South Africa. Now, again, what is the aim here? The aim is very simple. That is a significant reduction in current rate of biodiversity loss at global, regional, and local level. Again, aim is very important. Year is very important. Location is also very important. Then we have the Fleming chain and flora.
Yes, students, microbes chapter, Fleming chain and flora, very very important because they were the ones who got rewarded Nobel Prize for finding antibiotic potential. That means the actual potentiality of your antibiotics was found by Fleming chain and flora in the year 1945. Easy to remember, so do not forget. Again, a very small thing, but sometimes when we study, we actually skip it. Okay. Again, don't forget FCF. Now, students, some of the years. First is 1990. You have HGP, that is your Human Genome Project, a mega project which took almost a lot of years because in 2003 it was HGP project was completed. So around 13 years of period. This was continuing almost 13 years. So if the year is asked, like when did HGP start, initiate, that is in 1990, and when did HGP get completed was in 2003. So almost 13 years it took to complete this mega project. Okay. Now, very important, I saw this question a lot of times, but students also ignore this. In May 2006, the sequence of chromosome 1 was completed. That means like near future, right? I mean near past, right?
Near past.
Okay, near past. 2006 is like, I don't know, maybe your siblings are born during that time. In 2006, May 2006, the sequence of chromosome 1 was completed. So it's important for you to remember. They will ask you which is the chromosome, which is the chromosome that was found very recently. Okay, the sequence was found May 2006. Now, 1938, evolution. In evolution chapter, your coelacanth fish That is the first person who was like, okay, I should not be in water. Maybe I should be an amphibian. Okay, I should just go to land.
Exploring new horizons.
Yeah, just jumped out of the blue and then decided, okay, chalo, today I am going to write Neet 2024. May 4th, some people will decide. Okay, now that I've registered, I have to go. Okay.
Then we have the 1951 Family Planning of Program, that is
your
RCH. 1971 Reproductive Health Legalized MTP,
Medical Termination of Pregnancy.
1981
AIDS was first ever reported. Okay, first ever reported. 1983 students, your human insulin out of slaughtering cattle and other things, they were not able to find. Finally, Eli Lilly company, an American company, finally found that why should we do why should we do insulin, in the pro-insulin state, let's do it in the mature insulin state where they used only A peptide and a B peptide and completely remove the C peptide. Okay, very important question again. Eli Lilly, it's in the year 1983. Then 1997, Rosy cow, that's your transgenic cow. To add alpha lactalbumin in the milk, we found this Rosy cow, transgenic cow. That means basically.
One more was there, no?
Yeah, Tracy. Okay, Tracy, that is for alpha antitrypsin for emphysema, because emphysema, people who smoke, that will be less alpha antitrypsin. Now, sir, here, what I want to tell, transgenic animals, gene manipulated. Okay, genes are manipulated.
In 2022, ma'am. Really recently.
Very recently. That is basically, date is not mentioned, but biopiracy is when, sir has done some work and some multinational company takes it and does not pay him anything for it. Or does not give him any patent.
This is scam to me.
That's basically they scam, sir, like big time. Okay, that time we will use biopiracy.
Then in 1950, that whole plant could be regenerated from a plant. That is tissue culture.
Initial stage of tissue culture was found in 1950. That's when they realized that, okay, from a small portion of a plant, I can do a completely new individual.
Exactly. Right? Just take a small leaf from a mango plant, okay? Small leaf. In that small leaf, make some small holes. Okay, take a tiny bit of a leaf, grow that in your test tube with the proper media. You can actually grow an entire mango tree eventually. So remember that is the beauty of your plants. Animals, you can't do that.
Yeah, only that is left for animals. Okay, now 1990, the first ever clinical case of your SCID, that is absence of your ADA deficiency, right? ADA deficiency. The first clinical gene therapy was given in the year 1990 to a 4-year-old child with adenosine deaminase deficiency. Okay, yes, all of you are with me on this. ADA deficiency, very important because this will reduce the immunity if this is not there. Okay, now 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office when basmati rice was already found in India. They just modified a very little thing about it and they got patent, and this is how they led to biopiracy and multiple things.
I love basmati rice in biryani. The best combination.
Yes, here, gene therapy. Okay, gene therapy. Yes, 1990. That's all.
Do you realize, students? You might be thinking, sir, ma'am, you finished so fast. Yeah. So fast. Do you know how long does it take to make this PPT?
Yes. So, students, if we have missed any dates or any scientist because of new, textbook or old textbook, we will send you a PDF with all the dates and all the scientists. If anywhere something is missed, okay, you don't have to put it in the comments or anywhere. We know it, so we will send you the PDF. Okay.
And don't worry, I'll be doing the plant families very soon. I'll be giving you a quick rapid revision of all the plant families with very few points which you need to remember. Along with that, examples I'll be telling you. So don't worry, that is coming up very soon. Okay?
Yes. Jacob and Monod. Yeah, you can remember Jacob and Monod. Again, not very, loudly mentioned anywhere. But then when we teach, we usually keep telling about Jacob and Monod. Okay?
Lack of pronoun thing.
Yeah, lack of pronoun concept.
Yeah. So there it is, students. There's a link in the description for your mock test. Some of you were still wondering mock test, right? Where is mock test? When is mock test happening and everything? What do you say, Jima?
I'll just add Jacob and Monod.
So you can, we'll send in that. Otherwise, there are so many things written, right? Don't worry, Jacob and Monod will be added. Don't worry. So, students, if you have not started mock test, this is the last call because I feel we are actually planning to stop the mock test because many students are not taking it. Like many are taking it, but there are some not taking benefit of it. Yes. So we are planning to stop new entries from now.
Okay, now students, yes, AIMT completely shutting down on 28th. We hardly have 2 more days, no sir?
Yes, ma'am.
Yeah, we're stopping it on 28th. After that, no more registrations will be taken. And like I mentioned, yes, students, like we mentioned, there is huge surprises coming, and you will be able to meet all or see all of your favorite teachers of Vedantu Neet English on your Vidya Utsav that is happening on May 1st at 12 p.m. So quickly click on this link and just click on notify me so you do not miss any of this update. Okay.
Then we have your AI test series students. I'm telling you the last time. I'm telling you so many times. We are Shreyas have been telling, Gopika Ma'am have been telling, Disha have been telling. Please take advantage of what is given to you. Do not take it for granted. Do not be like, no, I will wait for until 4th May to write my first mock test. Do not wait. It will not be there. I'm telling you, it won't be there at this point. So right now, take advantage of it. Click here. AIMD test series. Click on it and you will have it for free. You don't have to see. Can you see? Enroll for now free. Mock test. 28th April over. It's gonna be. And then later on, I do not want any of my students to be, in difficulty or crying. Sir, can you give one more mock test, one more chance, one more chance? No, the chance will not come to you again. This is the final chance.
Yes, this is on a very serious note
Very
because
serious
we have
note.
been telling in very fun way, in other ways, multiple ways, we have been trying to put it in your head that without mock test, you will not crack NEET 2024. If it is just to mug up the theory, every single person can do it. Every 12th standard who got 99% also can do it. Right? Then why is it difficult? Because again, question practice. Okay?
So do it. This is last chance. You have today is 26th, right, ma'am?
Yes, you have only one more day, 28th. 27th, 11:59. Shut up.
Done.
Okay.
So if you have not taken your mock test, do it right now. Right now. This is last chance, ma'am.
Yes. And students, since a lot of you are asking us, ma'am, where is the link for that? Have you done frog? Have you done cockroach? Have you done this? Have you done that? We have a neat treasure box link. You can see.
See here.
Class
The students, you see here. Morphology, neat paper, as well as the neat PYQ, as well as the notes. Everything is there.
PYQ.
Everything is there here. Everything is here. Cell chapter, everything is there. Right? Some of the notes have not been updated, I feel, ma'am, still. Oh no, you will get it done. Yeah, notes have been updated here, yes. Here, students. This is for biology. Then we have physics. Every single chapter, as well as the NEET, as well as the class notes, PYQ, chapter-wise PDF, NCERT solution. And students, if you go to any of our videos, in the comments, first comment will be our own comment giving you the notes. So go and download the notes from there and let us know in the chat comment today. I want every student to comment today if this session was useful to you. If the session was useful to you, quickly like the video and consider subscribing to the channel, because all of this takes a lot of effort, and at the end of the day, when we see a lot of comments and we realize, yes, it was all worth it.
Okay.
Right, friends?
Yes, all of you are becoming conjuice in likes, comments. Now subscribe also. I don't know what to tell anymore. Okay, thank you so much, students. So we have finished the entire thing in 1 hour, which is amazing.
Exactly.
Okay, we have not wasted a single minute of yours. So if anything is missed anywhere, we will write it down and send it to you, share it with you. Do not worry, do not comment and be like, ma'am, you missed that, sir, you missed that. Okay, no, because when we go through many chapters, sometimes here and there we miss, because again, human errors can happen.
Exactly.
Okay, so yes, thank you so much. Love you all. Take care.